speaker, who is Lucie Bruijn. So Lucie has a very distinguished career in neurodegenerative diseases and MND spanning about 30 years. And Lucie came to prominence when she developed a SOD1G85R mouse model of MND, which is actually still in use by some members of this audience. So it's such a classic model. And this was followed by a string of papers in Nature, Science, Neuron. And I've always followed Lucie's career with enthusiasm as a PhD student. Lucie then became chief scientist and vice president of research at the ALS Association. And this is where she really spearheaded this global effort in translational research called Treat ALS, which you may hear about today. And Lucie's contributions underpin many of the current initiatives in ALS, such as the ATLAS trial, which is ongoing. Lucie is currently therapeutic area lead for ALS at Novartis in the UK, and she's overseeing development of drugs and biomarkers. Lucie has many honours, but the most distinguished recent honours was the awarding of the 2023 Humanitarian Award from the International Alliance of the ALS MND Associations for her groundbreaking research achievements and advocacy. Lucie really embodies research excellence, advocacy, drawing together partners globally in industry, academia, biotech, donors, and with hugely and sparking these worldwide initiatives. So I shall now hand over to Lucie. Thank you.
Well, thank you so much for that very kind introduction, Brad, and it's been wonderful to be with this community for so many years. And thank you for the kind invitation. It was such a stimulating day yesterday to see all the energy and the new ideas and the diverse ideas in research. So I'm going to probably cover many of the themes that you've heard yesterday and give a little bit of my angle in my different positions that I've held over time to give you a perspective of drug development, and will to some extent focus on biomarker development. Just have to see that to disclaim that I am a full-time employee of Novartis and will not be disclosing any specific Novartis programs, but give you a perspective of how we think about drug development here and there in the presentation. But I will start out with this image, and I'm not sure if Pam is in the audience yet. I haven't seen her, but her team now a few years ago wrote a beautiful review, and I think it's so nice to reference this. It's really what we talked about yesterday, and I'm sorry I missed Amar's presentation. I was on an ABC radio opportunity, but I think that the reality is, and he will have shared, the complexity of the many, many mechanisms that are going on. And here in this image, I highlighted all the different clinical trials and where they are potentially acting. And I think we probably, where we can be very optimistic, is that we are seeing successes, but we have seen a lot of failures. So I want to throw into it the themes of how we could do it better, perhaps, in what we've learned. Again, a classic image from Amar's older paper, and he probably gave you a more updated version yesterday. But I think it is the landscape of the number of genes that we found that really help us shape understanding of the pathways. And of course, the major genes, the SOD1, the first one, but then to follow TDP43, C9, and FUS. It's really through those kinds of targeted approaches that we might actually start to understand how to develop drugs effectively. And also the interesting biology, where we now know much more about the spectrum from ALS to pure FTD, and then those overlapping mechanisms and the genes that tie the two together. A really fascinating biology and why it presents in patients one way versus another way. So really the complexity is inescapable. So what are the challenges to drug development and to doing this right? Whilst we really have a growing knowledge and all the beautiful work that you're all doing here and the many models that we still have limited understanding in the human disease of which pathway is important at what time. And clearly, we're not going in early enough to give the therapies, but how do we do that? How do we get early enough? How do we know that the drug we choose to use is tackling the pathway at the right time? And you'll have the great debate. I already have my bias, and we've had a little bit of a taste of it at NCALS earlier about, is it targeted or is it cocktail? And undisputably, there are going to be many drugs that are going to be needed. But actually, I think it's going to be the development of one drug after another and combining with what's already approved that is going to be realistically what happens. And so how do we design these trials accurately to get the best out of them? Always remember, a clinical trial is an experiment, and it's a bit of a challenge to design the best experiment and do the best thing that we want to do for people living with the disease. So I think there are two major topics, and I've chosen to focus on one purely for time, is that really the better our preclinical work, our identification of the target we want to go after, be it whether it's in model systems, the better we do that, and we understand it in context of the human disease, the more successful we will be in our clinical study. And so the other very important part, and what I spend now my day-to-day thinking about, is developing those biomarkers for the clinical trial. So I'm going to focus on, I cannot go on a biomarker discussion without neurofilaments. We had a good discussion about it. I'll give my perspective a little bit today. I completely agree. There was a comment from Jeff that TDP-43 is our next opportunity. It might take long, but it's essential. I'll talk a little bit about some of the profiling work we've done internally, how we have to think carefully about standardizing assays, which I think the stage was set beautifully with neurofilaments. And if time permits, talk about a digital endpoint study we did at Novartis to inform us, and also the importance of engaging people living with the disease, all themes that we heard also this morning. And so, again, it was commented yesterday in the discussion about neurofilaments, the context of use of a biomarker is critical. And this is really taken from FDA, how they describe which biomarker you're talking about, be it diagnostic, prognostic, monitoring. So all of these things come into play. And when we're thinking internally of our clinical trial design, we think about the biomarker that's specific to our compound, as well as disease-specific biomarkers. And then also what I think is essential is that we think of biomarker development at the very time the team are thinking about which drug or which target they want to drug. And so I work closely with our preclinical teams and help the translation from these model systems into thinking about what the relevance is for humans, and then with our trialists in terms of bringing biomarkers into that particular clinical trial. And all of it is easier said than done, because a target engagement biomarker, it's not necessarily so easy. This is the ideal. We want one to show that we've engaged the target. We absolutely have to know that we have got our drug where we want it. So whether it's a biologic, whether it's antisense, whether the delivery method is oral or IV, we need to be sure that if we think it's a pathway that involves the glia or the astrocytes or the motor neurons, we have to get it there. Also important, of course, in what we talked about in context of neurofilaments is the treatment response. How do we know if that biomarker or if that treatment is shifting the pathway we care about? Do we have a biomarker that can highlight that? And then the next big leap of faith is, if we've shifted that, does that mean anything on the clinical outcome? And so all of these we have to think carefully about. Of course, a biomarker could, again, mean many different things. I'm talking mostly here about soluble biomarkers in the CSF or plasma. There are also other markers, the imaging ones that were mentioned, the limitations and challenges, potentially down the line, some opportunities, and then functional outcome measures that of ALS-FRS or any digital measures. And undoubtedly, you saw something similar from Amar yesterday morning, but this is our challenge. This is the ALS-FRS score, and each individual journey on the right, compilation on the left. And the challenge here is that it's extremely variable. Every person living with the disease can tell a different story. And so how, amongst all of that variability, can we then say that a drug is having its effect? And very interestingly, in fact, and this is, I think, an incredibly interesting area that's evolved, the story of UNC13, the story of how that links into TDP-43, these genes underlying can also change significantly the course of the disease. C9, we know, is generally could be a faster progressor, progressing population, and UNC13, if you're a carrier, can also cause a faster progression. And all of this then confuses or muddies the outcome in a clinical trial. So we heard yesterday how important neurofilaments are, and that it really is one of the first soluble biomarker that's been used in an FDA approval scenario, or at least a conditional approval. And there are many things about neurofilaments. I'm less concerned that neurofilaments are a marker in many diseases. I don't see neurofilaments as a diagnostic, with the exception, of course, of the cases C9 carriers and SOD carriers, where you can use it to determine that point of transition pre-manifestation of clinical symptoms. And so I think neurofilaments are really exciting and important for the field. And for those who are a little less familiar about what neurofilaments are, I was trying to explain it on the radio in a very quick way, and I don't know if I did that very well, but neurofilaments are a very, very important and component of the very long axon that connects our cell body to the muscles that these cells innervate. And so the whole architecture of that axon is made up with many things, but neurofilaments are very prominent: heavy, light, and medium chains. Actually, my very first project in John Cleveland's lab was to look for mutations in neurofilaments linked to ALS. And whilst it had been known as such a hallmark, you see in these dying neurons, these huge accumulations of neurofilaments, there are actually no mutations that directly link. There are some in neurofilament heavy that cause a risk propensity to ALS. So really fascinating. And so what really is happening, and Andrea Malaspina would be much more versed to give the details of it, but it's really, when you see neurofilament changes, it's an indication of this particular soluble marker being released in the CSF and plasma. There are many things that we have to think about, though, because if we're using it in context of inflammatory treatments, we know that neurofilaments might actually be elevated temporarily. And we also know with gene therapy delivery, because of the damage of that injury when you were first injecting the gene therapy, neurofilaments are also elevated. So we have to be very careful about how we read this out. It is, again, a testament to years and years of work that we've got to a point where we've got neurofilaments in place, and also the standardization that was done by many of you here in the audience. And so neurofilaments in clinical trials are certainly very exciting and important. They could become good stratifying biomarkers, determining faster and slower progressors. I don't think we're quite there yet, and it's a whole discussion about whether it's reasonable to pre-specify and to do things post hoc. I'm not the expert to explain that, but I would say when we're designing clinical trials internally, I do caution the team internally to say, look, we can't place all our bets on neurofilaments if our target is quite different to the target that was in the tofersen trial, and I'll highlight that again in a moment. So we know that neurofilaments are elevated, are more elevated the more aggressive the disease, and then they tend to stable out. The question really is when are we catching the neurofilaments in that particular patient? Are they still on the rise? So we have to be careful about that. But I think the best illustration of the success of this biomarker is in the Biogen trial, and we learned so much from tofersen. And here to highlight two important things. Neurofilaments take some time in themselves to be lowered significantly, and this was around the 2-3 month period. So we have to have a time frame when we predict when we might see that change in a biomarker. Is it going to be an earlier change? Is it going to take longer to change? And then just because that biomarker changed, it might not necessarily mean at that time frame the clinical outcome measures have changed. And we saw that very clearly here. And it's why actually tofersen was approved by the FDA only conditionally, because they didn't really have the data in the initial clinical trial design, the length of study to show them that, in fact, the downregulation of neurofilaments does indeed show a clinical outcome. And it's only because of their open label extension that they could surmise that, but their trial was not well designed to show that right away. So many things you have to think about carefully in that trial design. So to future biomarkers in the soluble space, and TDP-43, of course, for many reasons, is the one to go for. So interestingly, if we think of the whole biology of ALS or MND, 2% of ALS is caused by mutations in SOD1. And I do think we've made a huge stride. The success that we can actually treat 2% of ALS is a big deal, because in the past we might not have thought we could actually change the course of disease. And even more so now with FUS, younger disease course and FUS carriers, where some of us witnessed a person living with ALS who, from having to be completely supported, was able to walk independently. We used to hear those stories and knew that they were not true, and this was really believable. So on target, we can do amazing things, and we can actually make an impact in the disease. In fact, 98% of ALS, we have TDP43 involved. Not all mutant cases, TDP43 is part of the pathology. And I started my career actually in tau biology in Alzheimer's disease when everything was about amyloid, and tau was not so interesting. And in fact, we know now in Alzheimer's that there's two waves of pathology, and you don't have to have a mutation in tau for it to have a huge impact. And there are therapies really now focusing on tau. So whilst it's interesting that there are mutations in TDP43, it's not necessary for it to be a fundamental part of the biology, and many are working on this. So just to move quickly in, the concept of it is that we saw beautifully yesterday illustrated the concept that it's loss of function, gain of function, our challenges with model systems that they don't necessarily incorporate all of that and how we might address it. But I think the idea, the loss of function idea, and this whole new biology of cryptic splicing is incredibly exciting, because not only do we see this internally in the company as therapeutic options, but obviously globally we are thinking about it as developing biomarkers. And I know Bob said it will take years. I hope it won't take years. We need it so desperately. But of course, there's a lot of great initiatives and collaborations globally to put this together, and work done by Target ALS and partners have been really pivotal in this area. And so what it can do is two things. It can actually cause an early decay, and you get a different RNA profile, or you can get these peptides, which actually Phil Wong and Len Petruccelli and Michael Ward have now so nicely illustrated in patient samples and in the C9 population, one particular protein. What's really interesting about it, and on the graph here you see, is that this protein actually is elevated before clinical symptoms in C9, and almost takes a wave on earlier than when neurofilaments are low, and then it starts up. So actually, maybe combined, these could be a very interesting diagnostic biomarker in C9 cases for us to think about earlier treatments. But of course, we know that there are a growing number of these cryptic candidates. UNC13, very interesting, Staffman, some of them direct targets being used now for drug development. But can we use a signature or a pattern as a potential biomarker? So we're very interested in that, and working with partners academically to get them from EVs in CSF and plasma, where you might have a chance to get a more concentrated population, because TDP43 has been actually very difficult to develop in CSF fluid or in plasma. So in clinical trial context, we still don't have a TDP43 biomarker. So recognizing many in the field who've made pivotal contributions to this already. And this was an interesting clinical trial that I highlight, and it obviously brought attention to the team internally that this might be doable. There was some discussion about whether we really know the contribution in these so-called neuronal EVs. Is it really neuronal, and where it's coming from? But what they did in this clinical trial in collaboration with a company called Neurodex is they took these extracellular vesicles in which the TDP43 was concentrated. And in this study, I'm not really promoting or saying that this particular treatment modality is the one to go for, but it's a combination in which they combined two drugs, and they show certain changes, and they showed a change in TDP43. It hasn't been replicated, and I think there may be caveats, but it is illustrating that these could be a way to move forward with additional biomarkers. So in summary, I think that TDP43 has a long way to go, but as many companies, there's a real excitement to try to target them. TDP43, the loss of function, potentially the gain of function, the aggregation, lovely discussions about how TDP43 spreads. The Novartis is supporting Magdalena Polimendieu, who's doing some beautiful work in TDP43 spread. We just think this is something that we should, as a company, collaborate with with other companies and the community, because as we saw with neurofilaments, it certainly takes a village to make this something that we can bring into the clinic. So very briefly then, I'm not sure, I don't see a clock. How am I doing for time? Perfect. So one of the important things is, as I said, very early in whichever program we choose to focus on, is to build the concept of which biomarkers to move forward. And whilst I can't really speak at the moment about the trial that we hope to have started next year, and there'll be sites in Australia doing that, it takes quite some thinking and some concern about making biomarkers a stopping criteria early in the trial. Yes, it is a good idea to do that so that we are not forcing a very long trial in a drug treatment that is actually not working. But because we know so little about how these drugs are impacting some of these biomarkers, we don't want to stop a trial too early. So what we've done internally is try to use our partnerships in the academia to test these different assays and to see if we can really try to understand as much as we can in the disease population, and then translate what it might look like when we bring a treatment in. And we heard this morning about the MND Collective, and so I didn't highlight Australian efforts, but it's great to hear Bradley mentioned the work being done with large cohorts of patients here. Of course, Answer ALS and the work that Jeff did, Project Mine, and the great work from Leonard and the team internationally, and these platforms like TriCals. Industries like ourselves rely heavily on these resources, ProAct to model clinical trials, Niels to get samples. So it's just fantastic how the ALS community has such a plethora of these resources. So internally, what we did, we took a small cohort as a test cohort from the Niels MGH cohort, and we tried to see if we could replicate what had been published for many of the inflammatory biomarkers. Just very quickly to share the kinds of samples we had in hand, plasma and CSF, small sets. We also did an unbiased proteomics and somologics approach to look at all kinds of novel biomarkers. But importantly, internally, we had to be sure we were defining our clinical trial in the same way as done in all the trials, the fast, slow, and intermediate progressors. And then what we've built internally are these panels of biomarkers to multiplex and to be able to use smaller amounts of the CSF to do this. And the long and short of all of this work was to show that certain assays worked well for the neurofilaments, and we found we could compare CSF and plasma. We work with the Siemens platform, just standardized amongst many of the diseases. But that is encouraging that plasma and CSF for NFL is so consistent. Not quite the same story when we look at inflammatory markers. We really don't see plasma and CSF correlating very well. And we compared all the assays we did internally with a beautiful publication from James Berry and his team at MGH to reflect very much the same thing. And we certainly now have a panel of markers that we are going to work with to try to make the best decisions when we put our compound in place. So moving forward in the few moments we have left, I think that there's a lot of attention to this and there's a real benefit. And I think people living with the disease play a very big role in developing these digital outcomes together with us. And so at Novartis, we had a small study going on to really try to identify which of the digital outcomes are most meaningful in the clinic. And it's an interesting comments that came back from people doing this at home. The idea that you're seeing in front of you the disease progressing, so these multiple tests over and over, and some didn't actually really appreciate that it was so intensive and so often, because it really was a reminder of how it was declining. So there's a real balance to what a person living with disease would like to be involved in, and what is helpful for the trial. We looked at speech. We looked at, if we move forward here, it was set up in one usable iPad to be able to do all the different things, the voice technology, speech, the breathing, the timed up to go, which was the least favored outcome, and some drawing techniques that helped with the fine motor skills. And some of the learnings in terms of outcomes was that we certainly, like others, have shown that the self-administered ALS-FRS is very successful at home. I know there are teams, Angela's working with the community as well, to standardize these kinds of methods. I know for our trial, we're very pleased that a lot of the training is going to be done through the TriCals network for these things. And then beautiful work actually done by Jeremy Sheffer and others looking at speech. It's something that we've also thought will be very useful. and some very interesting fine motor work with the drawing skills. So I think a real potential, and something else I think needs to be standardized in the field so that we're using similar digital technologies that we can compare once we have the published data from different clinical trials. And then finally, to comment really on the importance of engaging the ALS community and people living with the disease. And I think some of the tensions are often in a clinical trial design to incorporate as many into the trial as possible. But then the challenge there is, are we actually getting the right patients? Is the disease too far progressed for us to be able to see a change? And so really our team internally has a dedicated team working with people living with the disease. I was excited to hear this morning the platform that you've developed. I know Rick Bedlack and Niels have got a fantastic learning center that really engages and both educates people with the disease about trials, but educates trialists about what it is that is important to those living with the disease. And I really believe that, for example, when we are internally challenged and the teams would say, well, it's not feasible to take CSF, so we can't have CSF biomarkers. I always push back and say that most centers are able to collect that. If you explain to someone living with the disease why it's so important, we would know and what it's going to give and what we can give back to the community as a whole. I think that these things are very possible. And one of the final things that we spent quite a bit of time, and a challenging time and good discussions actually at a TriCal's masterclass, is how long should a trial be? Of course, we want to get an answer very quickly, but I think I illustrated very carefully with the Biogen, the tofersen study, that in their 6 months, they would never have been able to find a clinical outcome, and they couldn't possibly get a drug approved. It was only because the neurofilaments were quite compelling and they could see that decrease in a timely way that we are where we are today. So we can't have short clinical trials. I understand why we want to make them as short as possible. And we as a company, together with our advisors, have felt that the sweet spot was around 9 months. But this was also something we engaged our people living with the disease around. And they really helped us come to that conclusion as well. So engagement of a community is really, really important. And I think we have a promising time ahead. And we just have to build the right trials with the right drugs in mind for the right target. And I think we will see more of the tofersens and those that we treat for FUS now. So thank you for your time and attention, and happy to answer any questions.
Great. Thank you, Lucie, for a wonderful overview of state of play in biomarkers and your innovations in this key area. So we've got about 5 minutes for questions. Who would like to start? We've got Luke in the front there.
Hi, thanks for the talk. I'm curious about TDP as a biomarker. Is there any thoughts on have you heard of the quaking-induced conversion assay that detects prion protein?
I don't know the assay, actually, no.
Oh, okay. So maybe we can talk afterwards. That might take a bit too long, but yeah. Okay, sorry.
Magda also did some work to detect the aggregates in maybe it's
an amplification assay, yes.
Yes. I do, yes.
Okay, cool.
Yeah. So the aggregates are just as good a potential biomarker as the cryptic splice sites, but it's always been quite a difficult area to determine where are the aggregates, which cell types, what is meaningful, how many aggregates. I've been in that aggregate world for SOD a lot with that same kind of conversation. So I think I didn't focus that much on the aggregates, but I do think there could be a biomarker, but what is relevant in which disease at what time is still very challenging. And now remember the technology.
Thanks.
I'm not the expert on it though.
David.
Thanks, Lucie, for the great talk. You kind of buzzed through it a little bit with the CSF versus plasma in terms of the inflammatory biomarkers. There are a lot of companies that are running trials right now looking at target engagement in plasma. Do you see anything in terms of, you said the difference, do they both respond in target engagement? Or if CSF is better, is that something that we need to be getting out there to make sure CSF is not better?
Definitely better.
Definitely better.
I was quite surprised that I didn't have a chance to ask at the talk at NCALS that Sanofi, and probably it was a practicality, that Sanofi did their RIPK1 study and only collected plasma. I was quite amazed. And in fact, they didn't really see a change in the biomarkers, and the drug didn't work either. But You have to go with CSF for inflammatory at this point until we really understand better. Martin Turner's done beautiful work with chitinase 1 CSF. It's just essential. And so when they said they were going to run the trial internally without CSF, I said, absolutely no, we'll get nowhere. You have to do it.
Because there are a lot of companies now that are just using plasma to try to look at target engagement, right? So how do we populate that out so that we don't end up being misled by what the results are?
Yeah, and recognize that it's challenging, but talk to the ALS community, the clinicians. They'll tell you it's not so impossible to get CSF. And it may mean you do have to do a combination of things. I mean, we ran a PET study talking about imaging. It's a different problem, but they weren't said every center couldn't do PET. So we had certain centers doing the PET and others doing the CSF. Ideally, you want everything done in every patient. But so there might be ways to design your trial where you're not reliant on every center collecting CSF. But you have to do it if you don't know enough about your drug.
Okay, thank you.
I've got a question for Michael Spetty. While the microphone's moving, seems to me that NFL is a good biomarker of lower motor neurons, given that it's reduced in SMA. So what about the upper motor neurons and cortex? How do we get a readout into that subset of patients in ALS?
I think that's very fair, and I don't think we have the ideal biomarker. And I think that we probably in trials don't only have upper or lower; we have a combination. But it's a very fair point that we have to be cautious of that.
Fantastic talk. There's been a revolution in the last 3 years in sports science for lipid analysis and lactate levels, such as Pogacar can now go up a hill 3 minutes faster than Armstrong, even without APO. And the thing is that we've looked at the lipidomics and what we've done, and it's the opposite effect of what Pogacar gets to a certain, once the patient's engaged. So it's a great possible measure of progression. And now you can do that from blood samples from the ear, because that's what all the athletes are doing. Is it worth extending there in terms of looking at?
Absolutely. I think it's a very good idea. And I think that's easily done in any of our clinical trials because we collect the samples. And so I very much liked your presentation, and it's something that I've noted as something worth doing, to look at the lipidomics and check. So thank you.
Question from Jeff.
Lucie, that was really fantastic. And I agree with everything you said.
Oh, thank you. I learned well.
A little focus. So this is a little more sciencey, a little focus on TP43. You threw up, and I know you have to go through things quickly. Phospho-TP43 is not the right readout, in my opinion. It's seen pathologically, but it's also seen in models where there's no loss of function. The key thing about TP43 is its loss of function. So then coming around to that NeuroSense study, measuring TP43 levels in the plasma also right now is completely worthless. It does correlate. There's actually a nice study in FTD. It was published in one of the prominent journals about levels, but not as a reporter for drug action. Because, I mean, you know this, and maybe some of the audience too, this is a protein that's nuclear. It comes cytoplasmic. Well, how that's reflected in any peripheral blood measure, even the neuronal markers, in the EV. I don't know what that means. And we've talked to the company about how stupid it is, quite frankly. But the functional readouts that you highlighted are really the more important readouts for target engagement.
I think it's interesting. I mean, that's why I was a bit cautious. We've also talked to Neurodex, and we're a little. What is interesting, there was recently the publication by Anya, I think she presented a talk with ALS, where she did look at the EVs to distinguish the FTD, the different phenotypes. So I think there's something there, but I take your point.
Do, but that's about sort of clinical phenotypes and higher levels, but that's not what many of us think about for TP43. Is it a druggable response? And how do we know if our drug's working? And so I think that's a different biology, and it will, I completely agree with you. You know, with all the stored biobanks for plasma and the background clinical data on those, we as a community, both academic and commercial, can really plow through that, I think, relatively quickly, and then begin to find out which are the 1, 2, 3, 10 markers that can be plexed into an assay. But I do think it's important for that target, which is heavily sporadic ALS, as opposed to not being SOD1 ALS. Still, I agree with you. I really think it's important.
I'm hopeful then that we will more quickly than the neurofilament world get something for TP43.
Well, I think a lot was learned from what Bob and others in the community, Mike Benatar, have done in neurofilament, and you can just plagiarize that and say, well, how do we do the same thing for TP43? Work with those clinical investigators. Thanks. It was great.
Thanks. Good comments.
Yeah. It's notes about biomarkers. Is that okay?
That's okay. Anything's allowed.
Very nice presentation. I agree almost with everything you said.
Not
But
required, by the way,
yeah.
you have to agree.
it's about the trial duration. I think 36 months is just, you're just trying to be nice and to make it a compromise between US and Europe. I don't like compromises in that sense, but so I think it's just going
Should be
towards at least 48. And I think if you talk to Amar, but he's too polite also. I mean, you have to even go to, what is it, 18 months you think is already short, is it? But I think we should stop this whole discussion about the duration of trials and go towards event-based design. We're actually doing with the TriMAC trial and the lithium study, so that you choose an event, could be survival or even another event, and then actually it's very common in oncology and in cardiovascular trials, and so you can use all the data from the patients that are in first, and you use it during with a longer follow-up. You can use all the data in the trial population. Have you considered that, using that?
I think that's so the challenge is how long will that first event take? I mean, so there is a balance between running trials in industry for 12, 16 months is really challenging. So do we think as a field we could do something that we can feel compelled by a 9 to 12 month trial?
Yeah, but so I think in terms of duration, we've done some modulation on that. Now, if you have a fixed duration design, you have to wait until the last patient in. You have to wait another 9 months before you can stop the trial. With this event based, you could even stop earlier. And I think if you modulate it, it's not so much longer than a fixed duration
I
trial.
think we should push forward to trying that out and encouraging. And I think the more there's interaction with industry teams around trial design, the better, because I think it's your influence of the data that will really impact an industry's decision on how to do it.
Yeah. All right. Thank you. Yeah.
Great.
Is that okay, Amar? Yeah. Okay, good. Thanks.
I think we'll leave it there. Great discussion, and please join me in thanking Lucie for her wonderful